While some studies have shown that white blood cells heroically restrict tumor growth and combat colorectal cancer, equally compelling evidence casts the white blood cells as malignant co-conspirators, bolstering the tumor and helping it spread. Now, new research clarifies the role of these intestinal white blood cells, known as grand T cells, in colorectal cancer. It turns out that the cells have a double-edged function. They rein in early-stage tumors but, as the disease progresses, undergo biochemical changes and switch sides, strengthening the tumor. The findings, published in Science, shed further light on the role of grand T cells in tumor growth, and may open new paths toward colorectal cancer therapies. Grand T cells that live in the gut act to prevent tumor formation, says Bernardo Reis, a research associate in the laboratory of Daniel Masita at the Rockefeller University. Composed of but a single layer of epithelial cells, this busy digestive region must absorb useful substances like nutrients, and reject harmful ones like foodborne pathogens, within a limited working space. Grand T cells mine the gaps, perpetually scanning the epithelium to maintain the integrity of the intestinal lining and prevent pathogens from invading the rest of the body. We had data showing grand T cells were protective, but the literature suggested that they also promote tumor growth, Reis says. We wanted to understand what these grand T cells were really up to. Working in a mouse model of colorectal cancer, Reis and colleagues derived grand T cells from the intestines of animals with early-stage tumors and from the tumors of mice with advanced cancer. In comparing these two sources of supposedly identical cells, the researchers were surprised to find vast molecular differences between them. For example, the two categories of grand T cells boasted different T cell receptors. Moreover, grand T cells that had entered the tumor produced IL-17, a cytokine that normally promotes inflammation in response to infection. The grand T cells had completely changed, Reis says. To confirm their findings, the team then used CRISPR gene editing technology to selectively remove T cell receptors from the white blood cells, changing the cells from anti-tumor to pro-tumor, or vice versa. In this way, they managed to increase the number and decrease the size of tumors in mouse models. When we depleted the original grand T cells, the mice became sicker, Reis says. And when we depleted the tumor-invading grand T cells, the tumor shrank. Hope for Human Cancers. Reis and colleagues witnessed similar activity in grand T cells derived from human colorectal tumors and their environs. Cells inside the tumor resembled the renegade, late-stage grand T cells seen in mice, while cells hovering around the outside of the tumor looked more like the original res. It almost looked like a fight between these two populations, Reis says. Future studies will delve deeper, examining whether it might be possible to modulate normal grand T cells to curb the tumor and prevent their cancer-promoting alter egos from dominating the arena. Perhaps we might one day fashion grand T cells into Trojan horses that can act as anti-cancer cells right inside the tumor microenvironment, he says. Make a difference. Sponsored opportunity. Story source. Materials provided by Rockefeller University. Note: content may be edited for style and length. (2022), July 22. Colorectal cancer tumors both helped and hindered by T cells. Science Daily. Retrieved July 22, 2022 from ift.tt/Winnis4U Rockefeller University. Thanks for watching. Please subscribe my channel for more videos.